I will start, since we're going to talk about molecular machines. I think I should spend at least a few minutes talking about natural molecular machines. So as a matter of fact, actually, nature has found ways to build molecular machines that can accomplish almost everything you could actually think of um, on the molecular scale, obviously. And that's great because we can mine nature. We can basically, do prospecting, bioprospecting, as it's called, in nature to find molecules that have sort of desired functions. And we can repurpose then these natural molecular machines and use them for some other purpose. And I want to show you some examples where this happened in the past and where huge commercial value has been associated with, molecule objects. And let's talk about the object here shown in the bottom left. So this is a schematic drawing of antibodies, of an antibody, IgG antibody. Strictly speaking, this is not a machine. It's more like a molecular device. So, and its function is to recognize another molecule with high affinity and high specificity. And I mean, there's a huge commercial value now associated with antibodies. There's a technique called with which you can make monoclonal antibodies and sort of produce them in large amounts. And they're nowadays being used as therapeutics, and as a cancer therapeutics and immunotherapy. There's lots of maybe even antivirals. You heard about this, I guess, also in the context of the current pandemics. This huge commercial value of the simple functionality associated with the simple functionality of recognizing a specific target molecule, and antibodies can accomplish this function. Another example here is shown on the bottom right. This is actually really, you can think of this object really as a molecular machine. It's a restriction enzyme. What does that mean? It's protein-based enzymes which can actually cut DNA or RNA. But this example is about is a restriction enzyme that can cut double-stranded DNA, and so they can recognize particular sequences based on the edges of these base pairs, and then cut the duplex DNA into a particular site. So that's great, right? So these things have been found in the 1980s, if I'm not mistaken, and they're they are at the heart of molecular cloning. And there was a huge contribution that these restriction enzymes made to the sort of modern day molecular biology, and are still making. Also genetic fingerprinting, like in forensic science, for example, you do this with restriction enzymes. You have a DNA sample, you cut it with restriction enzymes, and you get a particular fingerprint of fragments that typically unambiguously corresponds to a particular individual based on genomic sequence. So this is an example of a molecular machine. Or like a more exciting, or maybe let's say not exciting, a more complicated, or like an example of a molecular machine with a more complex function is shown here in the bottom, in the top left. It's a DNA polymerase. So this is a molecular machine that can make a copy of a DNA molecule. So it's really like an enzyme that actually makes new molecules. So the restriction enzyme basically was just merely cutting covalent bonds, and this one, the DNA polymerase, can catalyze the formation of new covalent bonds. Again, there's huge commercial value associated with polymerases. Every PCR reaction that's been done around the world needs polymerases, and especially right now in the pandemic with all these real-time PCR testing efforts around the world, there's a lot of polymerases that are being used right now in a technological context for diagnostics. And then in the top right, sort of the most recent example for a molecular machine, which is surely going to find a lot of use in technology. This is CRISPR-Cas complex. So this protein-based machinery, which allows you to site-specifically cut or edit genomic information. And there's also a way which this machinery can be used to detect nucleic acids. So there's also COVID testing assays which use CRISPR-Cas technology. They're known as Sherlock, a detector, and gene editing. All these things, you've heard about them. I'm sure there will be a huge market formed around CRISPR-Cas technology. So these are examples which are useful, and they help creating new types of products which actually have an impact, a positive impact on society. But in a way, these things have been found by serendipity, right? So like restriction enzymes weren't found basically were found accidentally in a way. So, if they hadn't been found in the 1980s, maybe it would have taken until today to actually find these kind of restriction enzymes. So that's intellectually a little bit unsatisfying, right? So, you can only find what actually is there out in nature. But if an object isn't there in nature, it will be impossible to find it, right? So what if you needed a totally different functionality that is very unlikely to occur in nature? So I'm going to give you an example of such a functionality. What if you needed a reverse translatase? What is a reverse translatase? Well, that's a hypothetical, a fictitious molecular machine, which can take any given protein molecule and reverse translate the primary sequence of that protein into an RNA molecule. So that's basically inverting the central dogma of biology. And I think if such a molecular machine existed in nature, it would have been found already. So since it probably won't be out there in nature, if we wanted to have this functionality, we would have to build it ourselves. So what reverse translatase will be awesome to have. I mean, it could basically mean you could take, you could profile any given biological sample and store the protein content in sequences. So, I think if you spend some time thinking about it, you will see that there could be a huge commercial value of this. It could be really useful, the reverse translatase. Another example which could be really useful is a protein polymerase, right? We have DNA polymerases and they're used in PCR. So what if you had the protein polymerase? So you want to detect a trace amount of a particular protein, for example, a viral antigen. If you had a protein polymerase, that would be another fictitious molecular machine which can take a target protein, unfold it, and synthesize a copy of the protein based on the protein primary sequence. So, and then you could exponentially amplify the target protein. So again, such a molecular machine doesn't exist in nature to the best of our knowledge. So if we wanted to have this functionality, we would have to build it. And the inconvenient truth is mankind cannot build such molecular machines. So, we're going to venture and say we cannot build molecular machines for hardly anything. And that's very intellectually unsatisfying. It's also unsatisfying from a technological point of view, right? There's certain things you would like to do on molecular scale, and we just can't because we don't know how to build molecular machines. So this is the background of the mission of our research, my laboratory, and of many other colleagues out there in the world. We like to be able to realize functions that are inspired by natural macromolecules, but not necessarily existing in nature. We want to realize such functions in synthetic macromolecular objects, synthetic macromolecular machines. And then, of course, we'd like to be able to integrate such functional modules into higher order systems, which have then yet more complex functionality. You could think of something like a molecular robot, which you could then apply for a variety of purposes in science, technology, and health. And for the rest of my talk, you will see various examples of perspectives where you could use such kind of technology in health, for example. So if you wanted to get there, we need to learn how to build complex molecular structures and machines. There's no way around this. And we need to be able to do this de novo from first principles. I want to comment a little bit on the challenges associated with this endeavor. So let's look at the length scales. of various types of machines that on the macro scale, macro scale machines, they're on the meter length scale, and then the molecular machines we're going to be talking about, we're going to be interested in super tiny nanoscale. Like the natural molecular machines, they have typically dimensions, protein machines on the order of a few nanometers. And if you consider a virus still a molecular machine, at least a molecular mechanism, then it goes up to the scale of 100 nanometer, and you will have tens of thousands or maybe millions of atoms that have been arranged in some defined way in these machines. So this is a challenge to traditional synthetic methods. So we can just synthesize these kind of objects using the methods of organic synthesis. We need to invent new ways to build such objects. And I think viable starting point would be to simply try to learn and adapt the methods that nature uses to build such functional macromolecular objects. And the basic principle is to encode structures in the sequences of heteropolymers. And in principle, if you're already adapting natural principle, you might as well also think about using natural heteropolymers. So then we would have basically two families here. We could think about using polypeptides or amino acid chains, and we could think about using nucleic acid chains. So depending on which branch you pick, we end up in the field that is known as de novo protein design. And if you take the right-hand branch, you're going to be looking at DNA RNA nanotechnology. De novo protein design is an extremely exciting field which has seen a lot of progress recently, or as David Baker coins it, has seen the coming of age of protein design. So I think probably the leading laboratory in the world is the Institute of Protein Design at the University of Washington, headed by David Baker, which is really like it's an impressive pioneer of protein design. And it looks like, based on what they're doing, they can almost build almost anything right now using amino acid chains. But there's a new kid on the block, a new player in this protein, in the protein business, and there's a website of DeepMind here. There's a little description of a new machine learning based protein structure prediction algorithm called AlphaFold 2. So what these researchers did, they solved the protein structure prediction problem, and that's sort of a necessary step to actually work on a protein design problem, because in order to design an amino acid sequence which will fold into a desired target structure, you need to be able to predict the target structure that a particular amino acid chain sequence will adopt. So this is and protein design is an exciting field to watch out for and to follow it. I happen to be working here in the other branch, DNA RNA nanotechnology, and I would argue, and I hope you will agree after you've seen the rest of my talk, that DNA and RNA nanotechnology also has its merits. So there are some practical advantages to working with nucleic acids. So first of all, long sequences are easily made with highly commercialized DNA and RNA synthesis methods. And from a physics point of view, you have a reduced interaction complexity. So when you work with amino acids, natural amino acid chains, you typically, you have basically a library of 20 building blocks, 20 different amino acids, and basically a huge variety of physical interactions that can take place between these amino acid rest groups. Whereas in the world of nucleic acids, we typically work with only 4 building blocks, ATGC, and sort of there's one dominant mode of interaction between these bases, which is Watson-Crick base pairing, which facilitates structure which facilitates the design problem, as we will see. So I will be talking for the rest of the talk mostly about our contributions to this field. So, but I want to point out a few examples of the recent literature in which you can get a comprehensive overview over the entire field of DNA nanotechnology. So first of all, I want to highlight this review here, which appeared in Chemical Reviews. It's from Hao Yan and colleagues at Arizona State University. I call this the mother of all reviews because it's huge. It's 57 pages, 28 figures, and almost 600 references. It covers pretty much anything that had been made up to 2017. So it's a good starting point to see the versatility of DNA origami in particular. There's another review which also has a little bit of a historic perspective. It's simply called DNA Nanotech by Nadrian Seaman, basically the founding father of DNA Nanotech, and Hennadi Sleiman. It appeared in late, by the end of 2017. So, since then, 3 years, now 3 years passed, so there's new things that came up. We actually also wrote a review recently. It appeared a year ago, a little over a year ago, in Nature Reviews Genetics. It's a little bit focused more on the prospect of building marker machines using DNA molecules. Maybe you'll find this one an interesting article. Then there is a review about programming structured DNA assemblies to probe biophysical processes. It appeared in Annual Review of Biophysics. In May 2019, it's really focused more on biophysics applications of DNA nanotech, written by Mark Bartha and colleagues at MIT. And actually, during last year, we wrote another review entitled Advancing Biophysics with DNA Origami, which will also appear in Annual Review of Biophysics pretty soon, I think like in the next 1 or 2 months. So, and it sort of will contain the most current developments. And hopefully you'll find this interesting. Okay, so we'll be talking about encoding structures in DNA sequences. I'd like to give you a brief primer on how that works. So if you have 2 DNA molecules, 2 DNA single strands with complementary sequences, what does it mean? Well, if you look at this, DNA single strand, you see there's 4 colors. 4 colors indicate the 4 different bases of DNA. And there's a certain rule with which these rest groups can interact. For example, red likes to pair with orange, and the green one likes to pair with blue. So if these 2 DNA molecules with complementary sequences meet in solution, they can wrap around each other and form what is called a DNA double helix as a right-handed structure. And you see here the base pairs. So that's it. Oh, is that it? No, it's not it. You can actually, this very simple concept can be expanded to make sort of higher order structures. So to give you like a very simple example how you could go about making structures using DNA molecules, let's look at the set of 5 DNA single strands. So each of these DNA single strands, could be designed to a particular sequence. So you could design the sequence such that, for example, the green molecule has a certain stretch which is sequence complementary to a part of the blue molecule. So like they form a double helical domain here, but it could also contain a segment which, would be what's great complementary to a part on the pink molecule. So it could form a double helix there, and then maybe a third section which would base pair with the red molecule, so it could form double helix here. So if you have, you set up a set of potential interactions by sequence complementary in a way that you could form, for example, this little rectangle with these, branching appendices. So that would be, a very simple structure. And basically, you can, by using simple base Watson-Crick complementarity rules, you can design DNA molecules so that multiple DNA molecules can fold into complex shapes. And the original concept really was invented by Nader and Seaman in the 1980s, and has since been expanded significantly in terms of the complexity that we can achieve. So nowadays we have various methods available, and please look into the reviews to learn more about the details of these methods, which allow us to come up with this with sequences for hundreds of DNA single strands through a rational design process. And as a set, all of these molecules can sort of be brought into the same solution, and they will self-assemble and fold into a target object whose shape is entirely specified by the sequences. So it's a self-assembly process, and it occurs because the target structure sort of offers the highest number of base pairs, and thereby, under right conditions, represents a global free energy minimum, and where the system will evolve toward. I have an illustration here of a particular design modality, which is become known as DNA origami, which was invented originally by Paul Rothemont at California Institute of Technology and published in 2006 for making sort of single layer structures, and has been expanded in particular ways. And in DNA origami, you basically you have one very long DNA single strand, which is indicated here in blue, and you have many short DNA oligonucleotides, DNA strands, which are designed to base pair and on different portions of this, what is on this blue strand, which is also known as a scaffold strand. So then you bring all of these molecules into solution, and then, you know, through diffusion, these short oligonucleotides, also known as staple strands, they can, you know, base pair with the scaffold strand. And as all of these base pairs are being formed, actually, the long scaffold strand and all the staples together, they will form a the target structure. In this case, and this example, which is just an illustration, it's a simpler rod consisting of multiple DNA double helices arrayed in a bundle here. So this was fiction, and now comes reality. So with DNA origami in particular, we can build nowadays a huge variety of structures. And what you see here are, I think, 47 different DNA-based objects that we designed over the course of the last 2 years in my laboratory. And everything that's yellow and blue is made entirely from DNA. And these aren't models or something. So what you see is actual data. So these are structures that have been solved using cryo-electron microscopy of these objects. As a way of showing you the scale of these objects, I have included here two natural objects. So here in the top, in the outer orbit, so to speak, there's this little magenta colored object. So this is actually a protein complex, 800 kilodalton in molecular mass, about, say, 8 nanometer in diameter. So, and then here in the inner orbit, there's this magenta colored sphere. This is actually a hepatitis B core capsid. And this is about 35 nanometer in diameter. It's also cryo-EM structure. So what this little illustration shows you, that with modern day DNA and nanotechnology, you can build a huge variety of shapes on the scale of, you know, that is commensurate to those of natural macromolecular machines. So this is a huge opportunity. Allows us to build scaffolds on which we can position functions, and that can basically then accomplish something that we as a designer want them to accomplish. Nonetheless, most of these objects in this illustration are actually just sculptures, and we don't want to be sort of artists that make sculptures that you can only look at if you have a sort of a fancy microscope. We want to be engineers who design molecular objects to accomplish particular functions, right? So, and ever since sort of I founded my lab, we have worked on, I think, reasonably systematically on the roadmap designed to deliver ever-increasing structural and functional complexity. So this is shown basically like a timeline on the various things that we worked on over the past years. And what I will do now, I will take you through some of the recent progress that we've made. I think in a good portion we'll also be talking about unpublished data. It's not yet been printed anywhere. And I'd like to talk about this aspect of atomically accurate design first. Okay, so as in any other field of science and technology, or like particular technology, right? Precision design needs precision validation. So you can only design as accurately as you can actually experimentally validate what you build. So we use cryo-electron microscopy to do structural validation in the lab. We're in the fortunate position to actually have pretty good access to one of these devices. So what you see here is a 300 kilowatt cryo-electron microscope of Thermo Fisher, known as a Titan Cryo. So that's like a very popular device, structural biologists. We use it to solve the solution structures of synthetic DNA assemblies that we designed in the lab. So I want to briefly illustrate how that works. So basically, we make objects in the lab. So then, as a result of that, making these objects means we have a solution containing particles, structures, our target objects at a particular concentration. So then we'll pipette this part of the solution onto a little transition electron microscope support grid, and this will be shock frozen to liquid nitrogen temperatures. And if you do this freezing quickly enough, the water basically doesn't have sufficient time to actually crystallize, so it will become a solid phase but amorphous. So you get a vitrified ice phase in which the particles, which were formerly in the liquid phase, freely diffusing, they're basically frozen in place at their orientations and locations that they had just prior to freezing. And the fact that the water is amorphous is very important, because then you can actually still look through the samples in an electron microscope. So the water would be crystalline, for example, it would just diffract and you wouldn't see anything. So, and then what you see here is actually an exemplary cryo micrograph acquired in such a shock frozen solution containing Gaussian objects. And you can see there's a bunch of particles here, which all look a little bit different. And this is because they're suspended in different orientations in the thin layer of this amorphous ice. So then you can go and use modern methods of cryo-electron microscopy to pick particle images and sort of use machine learning and other algorithms to sort through the data and figure out how all of these views through these particles might be combined into a common three-dimensional structure. So if you do this, then you get data which looks a bit like this. So you have a object in the center here. It's like a 3D reconstruction from the data set from many particles. And then the sphere, which you see here surrounding it, is actually a histogram. It's a three-dimensional histogram. It shows you the number of particles which have been associated in the data set with a particular viewing angle. So if you have enough data and all your viewing angles have been covered, you can actually get pretty decent resolution. So this is the actual 3D reconstruction then from this data set. So it's a 126 helix bundles, like a barrel-shaped DNA object. Total molecular weight is molecular mass is 12 megadalton. You can kind of see right here is some helical signatures, major minor groove, all the connections that this is pretty nice. So for a long time, actually, you got stuck at this resolution, at a resolution where you can see individual helices, but not much further detail. Only recently, we discovered that we really have to embrace the fact that these DNA structures have some kind of flexibility. So this conformation heterogeneity associated with these objects. And once we appreciated this fact, we came up with some methods to deal with them. Well, basically, we adopted methods which had been previously known in protein-based structural biology and sort of co-opted them. And then we could go to high resolution. So one thing that you can actually see in these data sets is that we have what we call lattice breathing going on. So what you see here is it's actually a movie in which a whole set of different structures which could be reconstructed from the same data set as had been superimposed. So this object is also what you see is known as the pointer, something which we published like in 2012. And sort of this lattice-based object. Here, the DNA molecules point in this direction. So, and the internal structure is, lots of these helices are kind of arrayed in a lattice. And we can see that sort of in this whole data set, we find actually an ensemble of structures which kind of differ a little bit in the lattice constant. So there's something that we call lattice breathing, which adds a heterogeneity. If you were to neglect this fact and you just, reconstruct the whole data set as if it would be one static entity, you will blur detail because all the objects are kind of a little bit different. The other thing that we have in these data sets is that we have something we call domain motion. So this is another object which has sort of a bulky stem. We're looking like this along the helical axis, and has some skinnier domains which can fluctuate and move around. So in the same data set, you have basically different particles which, due to thermal fluctuations and agitations, they kind of, they have slightly different shapes. And if you ignore that and just ignore the whole, reconstruct the whole data set as if you had one static structure in there, you're gonna lose resolution. So I'm going to illustrate this when I'm going to explain now how to deal with this structural heterogeneity. So what we do is first of all, we do pretend that the whole data set comes from one static structure. So we take all the single particle images that we have in this data set and use them to reconstruct what we call like a consensus 3D structure. This is something that you should see here on the left. It's not bad, but actually, it's really not bad. So here in this part, you can clearly see nicely helices, minor groove and major groove and connections, but there are also other parts of the object where you basically get very little data, signal. So here, for example, there's another domain which is actually as this long, and you're completely missing the signal, presumably because it's moving around, so it averages out. When you recombine the single particle images into one 3D structure. So what we do is we partition the target object into domains, and then there's something that's called, an algorithm called multi-body refinement, where basically you make first consensus map and you cut it up into pieces, and then you basically focus the refinement on these pieces, and let all the particles align relative to this piece that you're interested in. So we do here, for example, you have 4 domains, like the blue one, the red one, the green one, and this orange one. So we do separate refinements, reconstructions, focusing on these domains, and then we recombine them into a bigger map. And this is then, we call it a Frankenstein map because it's like kind of patched together. And now we have, much better signal in all the areas of the object. So if we look here, for example, the blue domain, now we can see the helical signatures up to the periphery of this domain. Here it was completely lost. The orange ones, maybe the difference is not so pronounced, but here in the red one, suddenly we can also reconstruct the entire, skinny 2x2 helix domain here. So this approach is beneficial. And then with this Frankenstein map, now we have basically the entire object dissolved in pretty nice detail. So the nominal resolution here is something around like 6.6 angstrom or something like this. And at this resolution, you can see, each helix with major minor groove detail. We can see all the phosphate backbone connections between the stable strands and the scaffold, which hold the object together. And then here you can start to appreciate the deviations from the idealist design, right? So when we look at this, so we designed this. I mean, of course, we didn't design it to be deformed in this particular way. We wanted to have a straight object. Also, you know, you look here, there's a defect here. Apparently, there's we maybe did some unwise design decisions here, which lead to a systematic folding defect. If you have this kind of resolution, you can look at the structure and refine it, right? So then you can go take this concept of multi-body refinement one step further with we call this scanning focused refinement. It's computationally a little bit intensive, but just going to explain the concept. So basically what we do is we first take the consensus map of the entire thing. Then we chop it up into the domains, something which is obvious to us as a domain. And then we take the domain separately and again further divide them into multibodies. So for example, you could take one of the bulky bricks and then say, I want to reconstruct specifically this region of the object. So you'll get high resolution here and less resolution there, because all the particles data will be sort of masked and aligned relative to this particular region where you're interested in. But then what you can do is you can basically scan the region of interest over the entire cross section and do basically iterative refinements in this peculiar way. And then patch the results from these separate focus reconstructions together again to make sort of a very, a high resolution Frankenstein map with more Frankenstein patches. So, and with this strategy, we can reconstruct quite some nice detail out of these cryo maps. So what you see here is a portion of this big object where you have, here is one DNA double helix, there's another DNA double helix, and here there's a strand crossover between them. And here you can see this little depression. So this is presumably a missing phosphate, right? This is a strand terminus here, and at the end of it, it doesn't have a phosphate. So this seems to be like a missing phosphate in the backbone of DNA. And you can kind of see here the granularity of the backbone. So we kind of see the phosphates. So then if you rotate this around, you can really see this is sort of, you can see here the single covalent bond that holds these helices together. That's pretty nice. And here's some other examples of this sort of, from detailing the structures, you can see really the bumpiness of the backbone. We can discriminate single strand crossover. So that's a single strand crossover from double strand crossover. So it's like two strands that make a crossover here, right? That's one connection, another connection with a little hole in between. We can see these kind of depressions of missing backbones at neck sides. And actually, which is kind of also was satisfying to see, we can go all the way to the periphery of these objects, where normally we used to have sort of the least resolution. And then we can see sort of the edges of the object. So here, there's a scaffold crossover at the outer interface of such an object. The scaffold goes over here, and then here is some density from poly T-tails, which are actually unstructured in principle, where they will be expected to be unstructured. So this kind of covers this peripheral crossover. So with this, at this level of detail, it becomes actually a little bit cumbersome to to look at these electron densities. There's a lot of detail in there, and it would be nice to really correlate locations in the 3D map with what you actually designed. And to do this, it would be very helpful to have an atomic model that sort of it's fitted into the electron density map, and it allows you to interpret the map, the data. So we also developed a semi-automatic algorithm for constructing such atomic models. And I want to briefly illustrate the general strategy of this algorithm using this movie. So this effort was actually led by Leah Feigl, very talented PhD student in my lab, who coded up this method. So basically what we do is we spawn an initial atomic model of our target structure using NGMD or OxDNA, the available structure prediction methods in the field. And then we, in the first stage, we sort of low pass filter our cryo-EM data so that it basically gives like a you know, a blob, and then we run an MD simulation where the EM density acts as an additional force field, pulling the atoms from of the DNA basically into particular places. So this method is called as MD flexible fitting, originally conceived by Klaus Schulten for atomic model construction with proteins, and we adapted this for making atomic models of DNA origami structures fitted into the experimentally derived cryo-EM density map. So this is how it looks like. So it's like, we call this cascaded relaxation. So at first we have a high resolution cryo-EM data, and then we successively increase the resolution of the cryo-EM. We release constraints on the DNA on the DNA model, and then to basically bypass some potential kinetic traps, and at the end you get a pretty nice fit, and then you have a pretty good atomic model that fits into the data. And here's another illustration movie showing this on the scale of an entire object. So that's the initial atomic model. There's a low pass filter cryo-EM density map, and then now you can see sort of the model is being rearranged, it fitted into the blob. The resolution of the blob is basically successively being increased up to the final resolution that the data then actually had. So that's how we make atomic models, and we have a whole series of atomic models now derived from data which is shown here. They've all been deposited on the protein data bank. I think in a single deposit action, we doubled the total amount of DNA coordinates in the data bank. And I want to just briefly mention these two objects here. They look very similar, but it's two variants of the same object. So this is the atomic model constructed from the first version of this object that we built here, and we designed it to study effect of global twisting, global twister formations in these square lattice objects. And you see there's quite some twist here. And then sort of we looked at the structure determined here, and then we did some rational design decisions to remove the twist. And we made a second variant of it, which then is, as you can see, which is much more orderly, doesn't have any defects anymore, and is pretty straight. And this is an example of how you can use iterative design and refinement to actually fix structural inadequacies and arrive at really something that you're interested in. So all of this, what I just told you, has been published recently under the title of Revealing the Structures of Megadalton Scale DNA Complexes, Nucleotide Resolution. So the paper has 20 cryo-EM structures and several atomic models. And this is great work by three very talented students, Massimo Cuba, Fabian Kohler, and Elia Feigl, and a bunch of other co-authors from my lab who contributed structures, materials, or just helped with analyzing the data. I hope you enjoyed this paper, and if you want the original data or like some designs, so for example, optimize some computational prediction routines, we're very happy to share everything. Okay, so if this basically with the method to experimentally test, to experimentally validate structures that actually have been formed, we can do these iterative design and test cycles now. But to be honest, this is a bit tedious. And also, I mean, not everybody has access to these high-end cryo-EM devices. So it would, of course, be much more useful to ultimately have an accurate structure prediction method in place, which you could use to sort of do, the whole design and predict. You do a design and predict process and basically in your office without doing actually any work in the lab, which is costly and time consuming, and only for the candidates which you think, will move further in your pipeline. Only for those you do a cryo-EM or another structural validation experimentally. And I think with the data set that I just showed you, we have a good chance that structure prediction algorithms that are out there in the world, like OxDNA, Mr. DNA, or the recent edition Snoopy, which I really like, from Seoul. So I think with the data set that I showed you, there's a good chance that there's enough constraints in the data set to actually accurately calibrate these prediction methods, so that we actually have then a set of structure prediction methods which work and are sufficiently accurate to be useful. Okay, and then what we want to do on in our side, since we're experimentalists, we want to learn how to further push the resolution. I mean, we're not satisfied with where we are right now. The best nominal resolutions we can get is right now something on the order of 3.8 angstrom or 3.7. We want to push this down to 2 angstrom, where you really can see the basis. And then we want to also consider new design approaches. So right now, most of the structures in the field are kind of regular arrays. You can think of this as log stacking in medieval ages. I mean, there are certain things you can build with this design approach. But what we actually want to do is build something, which is a more organic, irregular shape. Since we don't do this in the field right now, I took here a natural example to illustrate this. What you see here is a large subunit of the ribosome, which is a pretty fancy molecular machine. So we expect that this step is probably necessary to be able to realize molecular recognition or enzymatic catalysis, other fancy functions in these synthetic molecular machines. Okay, so at this point, we'll take a break and stop the recording, and I'll get back to you in part 2 of my lecture pretty soon. Thank you for listening.